Hello guys, welcome to Alankar's pharmacy classes again. We have discussed in our previous video we have discussed about bright field microscopy, dark field microscopy, phase contrast microscopy, and now the today's topic is fluorescence microscopy. So fluorescence microscopy, as it names itself, describe its principle that it is based on the image that is obtained by fluorescent light. So what is fluorescent light and how it is working, how it works, let us discuss it here. So the principle of fluorescent microscopy is based on fluorescent light. Now what is fluorescence light? That there are some compounds which absorbs UV radiation. That is the radiation of small wavelength, and they starts re-emit the light of higher wavelength of visible range. So that kind of phenomena is known as fluorescence, and that kind of components which cause such kind of fluorescence are known as fluorochromes. So idea is this: if we put the sample along with any dye or fluorochrome, then it will start emitting fluorescence light. And other than this, if we anyhow stop the light which is not coming from the specimen or sample, that means we will get the background dark and specimen illuminated with fluorescent light. So this is its idea. So the compound absorbs the short wavelength. First of all, the compounds which absorb the short wavelength UV radiation light will re-emit the light of longer wavelength, that is visible light. And for example, there are some dyes, there are uranium ores, and these are known as fluorochromes. And the phenomena is known as fluorescence. So the idea, again I am repeating, the idea is this: if the specimen is mixed with fluorochrome, any sort of dye, or fluorochrome or dye, that produces a fluorescence restricting the other light reaching to the eyepiece. So there will be a high contrast image with a jet black background, and the image appears in a fluorescent color. So that will be in this way we have increased the contrast of the image. The contrast is increased, and that's why there will be a more clarity in the image of the specimen. So for doing this, what we are doing here in fluorescent microscopy, we are using a special kind of lamp, that is a high intensity mercury arc lamp or xenon arc lamp, because we need a high intensity light beam. So let us discuss its ray diagram. So in this fluorescent microscopy, we are using a high intensity light beam coming from xenon arc lamp, or it may be a mercury arc lamp. So by using this xenon arc lamp, a high intensity light is produced, and that passes through a first excitation filter. This is actually the primary excitation filter. The work of primary excitation filter is to cut all kind of radiations, and it allows the passing through only blue light. And this blue light, when it passes through here through reflecting mirror and condenser, it is focused into the specimen, which is placed in the stage. So in this stage, the dye which starts illuminating fluorescent light. So see this fluorescent light, I have shown this in a green dotted lines. This fluorescent light is further, actually it is a mixture of light, is a mixture of a green light along with a blue light that is coming from here. So a green light, a mixture of a green light and a blue light, and it instructs to the barrier filter. Now, this barrier filter, the selection of barrier filter is such that this barrier filter is selected on the basis of what dye we are using. It may be of every dye is having their specific barrier filter. No. Here, what is happening? This light is the blue light and the green light together. They are coming upwards direction, and it passes through the barrier layer. And this what the barrier filter is doing. The function of barrier filter here is to stop the remaining blue light that is coming through the here, and it allows only green light to pass through. And this green light, where green light is coming from? This is coming from directly from the specimen or our sample. So in this way, how the image will look like? Image will look like will be having a dark background with a high fluorescent image of specimen. So this is actually the working of this fluorescence microscopy. So now let us discuss the application of fluorescence microscopy one by one. So the first and very important application of this microscopy is immunofluorescence. So what is this immunofluorescence? Immunofluorescence is a kind of an antigen antibody reaction, and in which we can determine the antigen that is present in the sample of patient serum. And how it works? In immunofluorescence, we are using antibodies that are specific for the antigen that we want to identify. Then these antibodies are labeled with any fluorochrome or a dye. These antibodies, when it is labeled with fluorochrome, are known as labeled antibodies. And these labeled antibodies, when mixed with the antigen, that will bind through its FAB portion to the antigen, and in this way, this antigen glows in the color of dye, which can be seen through this microscopy technique. So this is a first and very important application, and this is how we can identify the or diagnosis of some diseases can be performed. For example, syphilis, chlamydial infections, and the presence of rabies virus in the patient brain smears. So this is the immunofluorescence is one of the very important application part of this fluorescence microscopy. Now the next application, it can be also used to identify the Mycobacterium tuberculosis bacteria. So by using the Oramin O dye, it is or because of this Oramin O, the Mycobacterium tuberculosis absorb this dye and it glows in a yellow color. So we can identify this presence of Mycobacterium tuberculosis in this way. Similarly, Bacillus anthracis can also be identified by using fluorescent isothiocyanate dye. It glows in a green color. And these are another application, and another application, last application, that it is used to locate virus in infected tissue specimen using antibodies. So we take antibodies, and antibodies, by labeling that antibodies, we can identify the viruses also that is present in the patient sample. So these are the overall applications part of fluorescence microscopy, and hope you have liked it. So please be with me in upcoming videos also. We will continue our session in a continuous session in the microscopy. Our next topic will be a confocal microscopy. So please do like and please do share with your colleagues, and please be with me. Thank you all.